So now moving on to our second genus of gram-positive bacteria, the Streptococcus. So the last two videos in the playlist we're talking about Staphylococcus, specifically Staphylococcus aureus and Staphylococcus epidermidis. In this one we're moving on to Streptococcus. So it's another type of gram-positive bacteria, meaning it has a thick peptidoglycan cell wall and no lipopolysaccharide capsule. Streptococcus differ from Staphylococci in structure because they are typically found in a pair called a dimer, shown here, or in a chain, shown here on the top right, rather than in clusters. Because, like I mentioned when we were talking about Staphylococcus, the strep divide along a single plane, meaning say there's one single spherical streptococci. When it divides, it'll put another one directly to the right of it, and then it divides again, puts another one directly to the right of it, whereas Staphylococcus have this kind of grape structure where they just form clusters instead of chains. Strep divide on a single plane, like we just said, rather than radially. They are also facultative anaerobes like Staphylococcus. Streptococcus, on the other hand, do not produce catalase enzymes, and so they cannot break down hydrogen peroxide into water, so they're more vulnerable to the immune system, because you have immune cells like macrophages that use hydrogen peroxide to kill pathogens, and streptococci are unable to break down this hydrogen peroxide into water. A diplococcus refers to when there are only two cocci bacteria together, shown here. We can differentiate the different kinds of strep by a few processes. The first is serology, where we assess the reaction of antibodies to virally associated antigens. So, say there are different strains of Streptococcus, different species. The different species each have different antigens that they're producing, these different structures targeted by different antibodies. Let's say Streptococcus A has antigen A, Streptococcus B has antigen B. If we use an antibody that targets antigen A, and we have this reaction between the antibodies and antigens present in a sample, if you get an antigen-antibody complex, then we would know that there would be antigen A in the sample, and so we would know it would be Streptococcus A. Hopefully that made sense. The second one is Haemolysis, which we're going to kind of go over a bit more in the next slide, which is where we culture bacteria in horse blood, and we are able to differentiate different species of streptococci based on the hemolytic enzymes that they possess, because if strep have these hemolytic enzymes, they're able to break down the red blood cells in the nutrient agar, and we're going to be able to know that the strain that we're looking at, the species of strep that we're trying to identify, is a species of Streptococcus possessing these hemolytic enzymes. Another way we identify strep is under the microscope through cell arrangement. So, like we talked about before, they form the streptococci divide on a single plane, forming these chains. We also undergo physiological analysis from biological testing. So, getting into hemolysis, which might be new to you, I don't think we really go over it in A-levels or GCSE. Haemolysis testing involves culturing the bacteria on a petri dish in an agarose gel containing horse blood. Bacteria that produce hemolytic enzymes can be differentiated from those without hemolytic enzymes depending on whether they break down the erythrocytes, so the red blood cells, in the agar gel. A beta hemolysis, shown here on the image on the far left, is where the streptococci in the sample completely break down the red blood cells, creating a clear zone. So you can imagine you would have had the horse blood and the nutrient agar on this petri dish. They would have got the sample of Streptococcus or whatever from the patient sample, smeared it onto the petri dish, culture it for a few days, and left it and come back to it. And you can see that the red blood cells in the nutrient agar have been broken down, indicating that the streptococci have these hemolytic enzymes, and this is what we call a beta hemolysis. An alpha hemolysis is where these red blood cells have partially been broken down, creating a discolored zone shown in the middle. And then we have a gamma hemolysis where the red blood cells are not broken down, so there is no change in the blood agar medium surrounding the colonies. So that's it for hemolysis. Now we're going to move on to a topic called Lancefield groupings, which are a serological classification system used to identify bacteria based on the bacterial antigens they produce. So the Lancefield groupings give us insights into the significance of pathogens. We'll go deeper into Lancefield's groups A and B with regards to Streptococcus in a minute, well, probably in the next video now, but basically, let me see if I have a slides for it. No, I don't think I do. It doesn't particularly matter, you just need to know that Lancefield groupings are a way of classifying different types of bacteria based on the antigens they produce. So we're running out of time on this one, and the next topics are kind of dense, so I'll move that to the next video. So that's just kind of an introduction into the genus of Streptococcus and an overview of Haemolysis and Lancefield groups.